Hi everyone, I'm Fillipe Do Carmo. I'm here to talk about probiotics role in the gastrointestinal disease. Okay, the study show here was developed through a collaboration between the Federal University of Minas Gerais and the National Research Institute for Agriculture, Food and Environment. The collaboration between Brazil and France form International Associate Laboratory, or INRAE. The aim of this INRAE is to identify bacterial determinants involved in pro or anti-inflammatory process to characterize inflammatory process in disease and develop new treatments. If we do that, we use beneficial bacteria or compound. Okay, first introduction. Gut microbiota is diverse and complex. It is colonized by up of trillion of bacteria with a high diversity of species. The microbial colonization of human gut begins at gestation and birth, and immediately thereafter the microbiota changes. And several studies show the most of part of these bacteria are bacteroids and firmicutes. And this complex microbiota has immunological equilibrium, but when we have a disequilibrium of dysbiosis, we have an increase of pathogens, bacteria, and consequently we have an inflammation in the gut tract. You have intestinal inflammation. Gut microbes are key to many aspects of human health, including immune response and the metabolic process. We know that gut microbiome can influence a disease, but it's obvious that we can influence too. Diet and the medication have a strong influence on gut microbiota composition. And we can do the same thing using probiotics, because probiotic supplementation has several beneficial effects on human health. Okay, but how does probiotics do that? Probiotics can act through different mechanisms of action, like enhancement of epithelial barrier, increased adhesion to intestinal mucosa, inhibition of pathogen adhesion, competitive exclusion of pathogen microorganisms, production of antimicroorganism substance, and also modulate of the immune system. This is very interesting because a specific strain has one or more mechanisms of action to modulate the growth response, and we have a specific strain for a specific condition. In addition to realize these metabolites can act to regulate and mitigate inflammatory process, probiotic bacteria have structures that are implicated in response to the growth, like sulfase proteins, flagella, pili, and capsular polysaccharides. These structures which can regulate signalization pathway to produce cytokines, inhibit apoptosis, and attenuation inflammation, and enhance the function of gut epithelium. Okay, the presence and the identification of bacteria is compounds such as surface layer proteins can help us to select a bacteria or best candidate to a probiotic. And these surface proteins form a paracrystalline bidimensional array in external cell wall. And the surface protein may be involved in key functions in probiotic role, like resistance to extreme environment conditions like acid and biliary salts, bacterial persistence in the gut, adhesion to host cells, and immunomodulation. We know that S-layer proteins are recognized by dendritic cells and can stop inflammation in the host. Moreover, we have several criteria that help us to screen probiotic candidates, like assays like a simulated gastrointestinal condition, MEF and zeta potential that we study bacterial surface, antimicrobial activity, adhesion into intestinal epithelial cells, and also in vitro challenges in inflammatory conditions. These information can help us into in vivo models, like models mucocytes and colites. IBDs are characterized by chronic inflammation in the gastrointestinal tract. It is mainly present in two forms, ulcerative colitis and Crohn's disease. And ulcerative colitis is characterized by continuous inflammation limited to the colon and affects only the mucous layer, whereas Crohn's disease can occur in several segments of the gut. It can affect all layers of the intestinal wall. Another inflammatory condition is mucositis that affects a large portion of patients who undergo constant treatment with antineoplastic like 5-fluorouracil. Mucositis is a severe inflammation that affects the interdigestive tract. In the same time, the treatment for mucositis and colitis are ineffective and also cause a serious side effect. Some studies propose to use probiotic bacteria to help the treatment of mucositis and colitis, and one of these bacteria is a candidate. It's a Propionibacterium frenderichs, a gram-positive bacteria, safe to consumption, used as cheese, heparin starters, and they have a great probiotic potential in gastrointestinal disorders. And also, these bacteria have a surface layer protein. The surface layer protein is SLPB. This protein is we think it's involved in anti-inflammatory process. And we investigated the probiotic potential of Propionibacterium frenderichs. We selected 7 different Propionibacterium frenderichs strains, and we saw that different profile the adhesion to intestinal epithelial cells, 1829. The strain 129 is the strain of most of high potential to adhesion to epithelial cells in vitro, and this strain possessed a SLPB protein, and after knockout of this gene, we saw that a significant diminution of adhesion to epithelial cells. That implied that protein that's a knockout gene, it's involved in decrease of adhesion to epithelial cells. Moreover, the impact of SLPB gene mutation in Propionibacterium frenderichs 129 decrease complete and change complete the surface extractable proteins, surface charges like a potential zeta, and the surface hydrophobicity. And moreover, stress tolerance for acid and bile salt and health stress are completely altered. This implies that protein, SLB protein, is involved in several processes and several criteria that we use to select a probiotic candidates. To explore the effect of SLPDB gene mutation on immunomodulatory properties, we subject both strains, wild-type strains and the mutant strain, to in vitro challenge in 1829 cells stimulated by LPS. The relative expression of anti-inflammatory and pro-inflammatory genes are verified, and we saw that 129 strain, the wild-type strains, induced the expression of EL10 and the other inflammatory cytokines, but mutant strain lose the ability to induce EL10, and moreover, the mutant strains increase expression of pro-inflammatory cytokine like EL8. Moreover, we challenge both strains in vivo mucosite model induced by 5-FU, and we saw that ability of wild-type strain to mitigate inflammatory process caused by chemotherapy in mice, but wild-type strains lose this ability. And moreover, we identified that 129 strain it's able to enhance epithelial barrier. And in this way, it's evident that the SLPB protein has an important role in the anti-inflammatory effects of Propionibacterium friedreichii 129. So, a protective matrix like cheese, it's essential to prevent the protease. In this work, we see that the cheese prevents these proteases, and moreover, we see that SLPB protein it's able to induction only the SLPB protein is able to induction production of EL10. Okay, finally, we produce in industrial conditions an MHL cheese use the same strain, Propionibacterium friedreichii 129, in combination with other two strains, Lactobacillus and the Streptococcus, and we investigate the probiotic potential of the Emmental cheese in the SS coli models. We did pre-treatment of cheese, and after the induction of colitis, we saw that a consumption of Emmental cheese reduced severity of subsequent DSS-induced colitis inflammatory process, and we have mitigate histological score and the other parameters like disease activity index and weight loss. These are very important parameters for colitis disease. But the main question is, does this always work? No, unfortunately, no. We try to develop a functional food with other strain, Lactobacillus casei 01, and unfortunately, we don't have the same results we saw with Emmental cheese. For conclusion, the probiotic strains, one or more, needs ability to modulate the immune response and control the pro-inflammatory process, and moreover improve the epithelial barrier or modulate the microbiota as a specific strain for a specific condition. Thanks to everyone who took the time to watch this presentation.